So I'm going to explain a bit about the company. So the company is called RUBYnanomed, and what we do is we aim at doing non-invasive monitoring of cancer progression. So this will be the famous speech I told you about. Okay, this is slightly long because I want you to understand things. But I have already made a mistake when I introduced the startup. I said RUBYnanomed, and we do, and then I corrected myself and I said, well, we aim at doing. No, if you're going to speak to investors, you have to say what we do is non-invasive monitoring of cancer progression. You can't say we aim at doing. You know, you have to be confident. You have to say, no, we are doing this and we are going to do it and we are going to hand it. Like, we can potentially maybe somehow, no, I mean, you're going to do this, okay, because this is your company and you have to do it, because that's your idea and you believe in your idea. Okay. So, well, what's the problem we want to tackle? It's obviously, I mean, cancer. I don't have to say much about cancer. Maybe what I have to say is that we, at this point of the company, what we are tackling is metastatic cancer, because it's metastasis is the responsible of 90% of cancer deaths. Let me see, because I tend to speak too much. Okay, it's responsible of 90% of cancer deaths. So we dedicate our time to our goal to metastasis. I'm trying to pass the slide, but it seems like it doesn't want to work now. So we have to be a bit patient. Okay, there you go. So that's our problem. And the problem with the monitoring of metastasis is that when a person is diagnosed with cancer, so in the first diagnosis, what is done is a tissue biopsy, and according to that tissue biopsy, the cancer type is identified, and depending on the subtype of the cancer, there's going to be a better or a worse prognosis, and the therapy is going to be decided according to that tissue biopsy. Then, when this is removed and a person goes through treatment and they are considered cured, then that person can relapse at any time. And the way that relapse is monitored is with imaging techniques. And these imaging techniques, apart from being expensive, they are not done very frequently, right? So the first 3 years, they're every 6 months, and then the first 5 years every 6 months, and then after year 5, they're monitored only once a year. And so what happens is that there is usually a missing of the early spreading of cancer. Again, so metastasis. So we need to develop tools that can actually identify when metastasis is there earlier. So for that, we are relying on what is called liquid biopsy, which is about using body fluids, blood, saliva, urine, spinal fluid, to detect some biomarkers, in our case circulating tumor cells, which are the responsible of metastasis. So these are cells that shed from the primary tumor, they enter circulation, they travel along the body, eventually they invade secondary organs. So that's the metastasis, right? And so with the circulating tumor cells, CTCs, what we can do is non-invasive and early detections, because it's just a blood analysis. In our case, we use blood. We can actually give information about how the therapy is working, because depending on the number of CTCs, a person is going to be in relapse or in remission. So we can do progression of this monitoring. We can also develop technologies for companion diagnostics. Companion diagnostics, it helps to stratify population of patients, meaning create groups within a whole population of patients that might benefit from specific therapy or might not. So that's companion diagnostics, okay? Now, what's the challenge? We have a tool that can do this. What's the challenge? I mean, there is no demonstration of the clinical utility of CTCs. There are works that say, oh, person with more CTCs is in a worse condition and the prognosis is worse. But there is no clinical utility, meaning we cannot link that to a specific therapy, okay? The systems that are out there, the competitors, they are not sensitive enough, and we have a technology that is much more sensitive. And they cannot provide automated analysis. So this relies a lot on the user that is going going to do this analysis and on the lab that is being done. So we want to eliminate this sort of inter-user and inter-laboratory variability, and we can and we can also provide more information than only genetic information, because CTCs are alive in circulation and they're responsible of the metastasis directly. So what's our solution to overcome these challenges and this problem of not being able to do a proper monitoring and not being able to have a robust system in different labs? Well, we have developed this chip, as we call it, the Ruby chip. This is a chip based on microfluidics, and it's very simple. You just input blood in here, and the blood will exit through here. What you have is a microfilter done with micro and nano fabrication inside here. And so the blood passes through, and the separation of the filtering units in there has been designed in a way that it allows the healthy cells to pass through this filter and only to trap the cancer cells. And once we can trap these CTCs, then we can do full prognostic information on the patient. And we are not only doing the counting of these cells, but we are analyzing the phenotype. So the phenotype is highly related with the aggressivity of these cells. So that is something that it's still not being offered to oncologists from something that is approved by regulation bodies. And that's what we want to do because our technology can do it. So what's our proposition? Also, we don't need to prepare the blood. You can just process the blood straight away, unlike other competitors. We have, once we get the draw sample, once we draw the sample from our patient, we just insert it into the chip and we can have result in 3 hours. Our isolation efficiency for the circulating tumor cells is higher than 70%. And this might not sound amazing, but this is 8 times better than the only approved system, because it's highly challenging to isolate CTCs, because the number of CTCs in circulation is extremely low when compared to the healthy cells. So this is like finding a needle in a haystack, right? And we actually have a very efficient strategy for finding that needle, much more efficient than any other competitor out there. It's very selective, and it's cost efficient because we can sell single test for 300. So we have this patent that we submitted in November 2016, and now it has entered last year national phase, and we have filed national phase in the US, in Europe, and in China, which are the markets we have identified to be the most powerful. This is just an optic bright field merged with fluorescence image. So you see here the microfilters. So these are like columns, micro nanofabricated columns with a specific gap. And these big chunks, they are the cancer cells that then we analyze under the microscope, and then we emit the report. So with the different proof of concepts we did in different types of cancer, breast, prostate, colorectal, and so on, we have demonstrated that we can detect metastases 6 months up to 1 year earlier. And well, I mean, we all know that early detection is very important to be able to apply the therapy as soon as possible, to be able to save the most lives possible. So really, our drive is not to make money. Our drive is to put this in the hospital, because we all know we are surrounded by people that have cancer, and when some of my relatives had cancer in the past years, I wanted so badly to have this in the hospitals, because I know it works and we have it in the lab, to be able to monitor whether they're doing good or whether they are doing bad or whether the therapy has to be changed. But we can't do it because it's not ethic, because this is not approved. So that's what we want to really do. I'm not going to go into this. So this is what we have to achieve, an automated thing. So we have the chip, and now all the different steps, like getting the blood, processing it, doing the microscopy. This is, let's say, in steps that are separated from each other. So we just want to assemble a machine that you just need to put the chip, put the blood, you just end up with the full report. Okay. Now we have competitors, of course, we have competitors. The most important is cell search, has been out there for almost 10 years. It is the only one that has approval to be used in the clinic, in the hospitals, okay? So what we did is to compare our performance to the only one that is approved. And so in yellow, you see the number of CTCs that cell search could identify for different breast cancer patients. And in blue, you see the number of circulating tumor cells we could identify for those very same patients. You see the difference is huge. What does it happen? There is a threshold established here, which is above 5 CTCs. That person is in relapse and the therapy has to change. What did it happen with cell search? Most of the patients were considered in remission, so they were not given a therapy. But with us, most of them showed more than 5 CTCs, see all this big population. But those ones were not treated, the treatment didn't change because they were considered in remission by cell search, the only one approved. What does it happen that after a while, unfortunately, most of these patients passed away because there was not a heads up on time that could tell the oncologist, hey, this patient is actually not doing well, change therapy or look for another therapeutic strategy. So that's why we really want to be out there and in the hospital. Apart from that, we have all the assets, but I believe it's not important now, and I believe I've been speaking already for a bit. So we need to overtake competitors. Apart from these famous cell search, only one approved, there are many other companies in the liquid biopsy field doing RUO, and RUO stands for research use only. And so these are the two most important ones. These companies have, as you see, much lower efficiency than we do, and they are not able to do automation because of the way they do the isolation, because we use microfluidics, micro and nano fabrication. This allows the automation, allows the fast analysis, the not processing of the sample, and the interlab and inter-user non-variability. These two companies have been invested in the US and in the US by more than 10 million. You know, so that's the difference. When you are in Europe, you have much more difficult to convince people. So you have to take that into account as well. But we are happy with this. We are happy that I mean, we are happy that we can show that we are better. So then you have to show, you know, your unique business innovation, right? These are all big words that they use in entrepreneurship. So now what we want to, so we are in the research arena with these companies, and we can sell for research use only our chips. Doesn't need to be certified for that. Then we need to achieve certification, and for that you need to go through clinical trials, which are extremely expensive. We're talking here from 2 to 5 million minimum. In Europe, 20 million if you want to do it in the US. And then we want to jump to the next market segment, which is companion diagnostics, which is to sell the company to pharma so that they can use this chip to analyze which patients are going to benefit from specific therapies that they do develop. So what we really want to do is to unlock a new market in here, which is locked due to the limitations of the other technologies that are in the market. Well, the market is huge. I mean, the market size, right? The amount of money that this market is moving around, and we fall into different markets. Cancer diagnostics, 210 billion euros, obviously. Within cancer diagnostics, circulating tumor cells market, 22.5 billion. I mean, to me, sounds quite well. And then 7.6 for companion diagnostics market. So these are the different markets we are targeting. And then what's our business model? I spoke before about a business model, right? So it's very often in these sorts of companies that want to put a machine in a hospital to run blood of patients and see if they're doing well or not, to monitor cancer, to put the machine in a hospital for free, like on a rent, on a lease contract, and then only charge for consumables. So you set up a contract with the hospitals for a minimum amount of consumables, and so that's how you embed the price of your machine into the hospital through the amount of the chips, and the margin is actually quite high, 74%. So this is our business model. It's called razor blade business model. And now, how are we going to commercialize? So right now we're here. So this 2024 shouldn't be here. Sorry, this is because I didn't expand it properly. So we're in 2020. We're collaborating with 10 hospitals in 4 different countries: in Portugal, Spain, Italy, and Slovenia. Now we have done more than 1,000 tests, but in the research use only framework, and we have above 1,800 pre-orders. Um, how was this kicked off? Did we decide to do the startup? Because we had a research project with Professor Luis Costa, who is a very important oncologist here in Lisbon, in Portugal, and Professor Luis Costa was using our chips in the framework of a scientific project. And we said, hey, I need 1,000 of this because I want to do this study with my patients. And we're like, what? I mean, we can produce 20 at most per week. I mean, to get to 1,000, that's going to be very difficult. He said, well, then create a company and sell them. And so then we created a company in 2018. We were, pushed by customers, because they had a need, and we couldn't do the production in a fast pace. Uh, so, well, we aim at entering first the breast cancer market, because we have more proof of concept studies with breast cancer. We have others with colorectal, with melanoma, with prostate, but we're much more advanced with breast cancer. It's also a very big market, and also the only one approved. The cell search I told you, it is approved for breast, prostate, and colorectal. So there is a path for regulation approval, which is head-to-head comparison to something that is already approved. So that's a strategy to get a faster certification. This we already spoke about this, but when you talk to investors, you need to show them that by 2030 we are going to have 68 million euros. This to me was the most difficult thing to do, because these 68 million euros, how do you reach this number? Well, you reach this number saying that, oh, in 2022 you're going to enter these and these countries, so you're going to be selling to 20 hospitals. Then in 2023 you're going to be selling to double the hospitals. 2024 you're going to be selling to 4 times those hospitals. And I was saying, but how do you know that, right? When we were speaking to our financial mentors and to guys that were doing the economics part, and they were like, well, you don't know it, Sarah. There are projections. I was like, but I mean, do you have to base your projections in something? So you have to understand that this doesn't work with the equations that we use at the lab, and you have to understand that you have to make a set of assumptions that it's usually difficult to assume by a scientist, and you have to do it. I mean, and you can create different scenarios, like a more optimistic one, a more conservative one. We have done a very conservative scenario for the number of chips we're going to sell along the years, right? But this was something that also forced me to make a change. This is just, I already told you. And this again, you have to speak about the company growth. It's also quite important to tell them what is the staff growth, right? So we are not yet in 2021, we are 2020. We are, well, this year, because we're a very young company, as Reyes said, this year we made our first hiring. So we have the first person 100% hired by the company. So the 3 co-founders, which is Lorena Dieguez, who is the group leader that started this project some months before I arrived to INL, and Paulina Piairo, who is an oncobiologist, and myself, who am a nano and microfluidics technologist. We the co-founders, and we work for free. I mean, we do this in our over hours. So I have my job as a scientist, and then I do this not in my free time, right after work. So this is how it works at the beginning. It's a lot of work, but the things that you learn and the change of mindset, it's absolutely worth. And then if your institution is nice, maybe they can let you work 20% for the for the startup, 80% for the science, and there are these sorts of agreements that you can push. And well, we got some funding this week, and actually this week and a month ago. So we are going to have four people fully hired by the company. A major, let's say, changing point in our company was when we did hire a CEO, Chief Executive Officer, a boss. I mean, a person that really knows what's going on in innovation, and that has connections, and that has been working in companies for many years. And we were at the beginning like, but we don't know how to pay this person, because we're winning awards, but very small awards of 10,000 euros, 15,000 euros, 5,000 euros for the company. But one of these CEOs with experience in bringing startups to the next level, they charge you 5,000 euros a month, so minimum. Okay, so we had to first create a relationship with somebody we liked, we trusted, and we thought it was the right person. We've been mentored by him for 1 year, paying like for 10 hours or things like that. And then when we thought that it was a good match and we got a bit of funding, then we paid him part of the month and we gave him part of the company. So sometimes you are like, how am I going to hire a CEO? We are very early startup and we don't have the money. Well, there are other ways of paying. So you can give part of the company to people that you want to work for you. So, well, I mean, our financial history is that we did incorporate the company in 2018, but of course, some of these results were a huge effort that was invest from public competitive funding, which brought us to a good level to then create a startup, and we've been winning some awards in different acceleration innovation programs. This is nice for an early startup to go to some of these, because you learn all the things I told you about: solution, idea, patents, blah blah blah. So that's nice, and also it gives you a bit of money to keep going at the beginning, like 10,000, 5,000, so that you can create a landing page, a web page, so that you can pay your travels to go to health fairs, so you can create some flyers, these sorts of things, until you get the real funding. We're very happy because between last year and this year, we raised almost 1 million through different initiatives, some public, some private ones. So we are aiming at getting the next round of funding of 2 million in 2021 to be able to do clinical trials and put our system in the hospitals in 2023, hopefully. And well, I mean, this is the team. So we have support from INL as well. So these are the people. So Lorena used to be the CEO, and Enrique the business officer, but it's swapped. But I have to say that depends to the program we submit the application, because sometimes they give funding for women CEOs, because we have to, we have to rock it, and it's not as easy as for men, unfortunately, and myself and Paulina. Very important to have advisors, to have people out there in pharma, diagnostics, oncologists backing up your product. So we have these mentors from the Health Tech tab. You can access these for free through the European Technology Platform, which is an initiative of the European Commission. and you can sign up for the health tech tab program. You can, if you have an idea, or if you just want to explore how this is, and you are assigned individual mentors, which are usually from pharma and diagnostics companies, very big companies with a lot of experience and very nice, and then people from entrepreneurship. So this was the start of the company when Luis Costa said, RUBYnanomed will help us stratify our patients and personalize the therapy. We need it, and we need it now. And that's when we started everything. And well, I mean, what I want to say is that we are doing this in Portugal in the interface with Spain, because I'm Spanish, Lorena's Spanish as well. And we have found that the conversation with Spanish or Portuguese investors and funds, not always, but in general, it's not easy. They're very reluctant. They don't know how to do it. They're not well educated in science, and so we have to do this pre-education on what is it liquid biopsy, and what is cancer monitoring, and how do you do this, and what is it microfluidics, and you have to do all this work, right? So science awareness. um, you know, we in Spain very much need a wealthy innovation arena. It's improving, and there are many acceleration innovation programs in Spain. You just need to find the right one and spend a bit of time in exploring, if you're interested, which one. Because if you go to a generic one, if I said, you run the risk of competing with people developing apps for the phone that are going to just do an exit, which is selling the company, do an exit in 6 months for a lot of money. So there's no way you can compete with that. You can always reach out. You see other people that participated in previous year, contact them through email, through LinkedIn, and say, hey, I see that you participated in this innovation program. How did it work? Did you like it? Did you didn't like it? I mean, just don't be afraid. Um, in Spain, as we very well know, and this also happens in Portugal, and I feel a very much of a difference because I did work in the UK for almost 4 years. The decisions take a long time, and usually you're lost in bureaucracy, so that's difficult. You have to be very resilient to deal with that. um, Again, we have to educate them, and I think that the most limiting thing is that there is a very poor communication between academia, universities, research institutions with private companies. And that's we don't it's not fair if we say, oh, but they don't want to speak to us. I mean, it is fair, sometimes they don't want to, but we have to do our part first, which is science awareness, and we have to educate them, and we have to go out there, we have to try to speak to them. Sometimes you get surprises and they speak to you. We have to rely more in the technology transfer office of the universities as well, because they usually do a good job, and they are underrated. So we have to do that, and to start going out there. I mean, we are very we're very innovative in the Latin countries, I have to say, right? Because sometimes all these craziness and oh, let's go, let's do, let's sometimes it's helpful. So let's take advantage of that, of our creativity as well. So I think that's it. The most important thing I want to say, and this is the take home message of my last slide, is surround yourself by excellent, bright people, by exceptional people. And so this is my last slide. This is Professor Chris Abel. Chris was my supervisor in Cambridge, a great mentor, great supervisor. Chris passed away this Monday. It was sudden, but I want to take this opportunity to pay a tribute to Professor Chris Abel, because he was an exceptional person. Apart from being professor at the University of Cambridge, he was a serial entrepreneur. He created more than 4 spin-off companies. At the same time, he had 2 research groups. This spin-off company was sold by 1 billion, but still he would always find time to go with you for beers, to organize group activities, to go out to the nature, and so on. And then he didn't only have an impact in academia and in entrepreneurship, but also he created the first postdocs affairs committee at the University of Cambridge to give postdocs the same rights than PhD students. He created the Cambridge Zero, which is an initiative which aims at Cambridge being zero carbon footprint in 2030. And he did create the Cambridge Enterprise, also company. So surround yourself by people that ignite you, that motivate you, and that are bright. Talk to them, and that's how you can be successful. And I spoke a lot. Thank you very much for your attention. I'm sorry that this couldn't be in person, so we could have interacted more. But that's it. That's it. Thank you.
Okay, thank you very much. Thank you very much, Sarah, for your very nice, encouraging, and inspiring talk. For I think for the students and for everybody. I
Thank you. I
think
hope so.
tell us the keywords of. I see the thank yous from the students written. The keywords for the innovation, also the difficulties in the market. You also
Mhm.
saw,
Mhm.
I think, the very important that you have to have clear data, consistency in your experiments and so on, which is very, very important for science. And of course, at the end, the passion and that you want to do the things is the very most important, because if not, you don't do anything.
Yeah.
This also from my
Absolutely.
side. So first, let's see if the students, they have any questions. Billy, you have questions in the Faculty of Science?
Uh, I'm trying to ask students, uh, do you have
Or
any
anyone?
question for Sarah? No curiosity?
They are shy. It was very long. It's not, it's not one to one. I understand.
That's not the same, huh? That's not the same.
It's not the
Yes,
same.
for sure. Don't be shy. Don't be shy.
I have a question, a curiosity, because one thing that it was it surprised me, no, is when you talk about the investors and how you speak to investors here in Europe and in America, that you say that here they just want the numbers and they don't ask more for the science and the passion and so on. And what's the other side in US? Do you think that this is maybe because of the case that you explain us about Terranos, that they want to know more about science deeper to avoid these problems, or because thinking in the characters of how we are in Europe or in US is quite surprising for me also.
It is indeed, and this might have been my experience, right? But I spoke to other people; they also realized about this. I never thought about the reason, but it's actually it could actually be because of that, because of Theranos. I think that's quite plausible, and yes, indeed, it's surprising because they're usually more money oriented than in Europe. Just it's something cultural and historic. So it could be, it could be. Then it's also true that um, we had conversations when we went to the US with people that was working in companies exactly such as Pfizer, Bayer, Merck, etc., etc. But people very high in the hierarchy that were more supposed to be managers, but they knew what we were talking about, and they had very sharp questions about the science, and that struck me. It wasn't one or two; it was most of them. So yes, it could be a mixture that I really want to understand because I don't want you to trick me. Um, and it could also be a matter of culture because they speak about innovation, science, and so on much earlier in time than here, which is another problem. We never speak about innovation to students, so almost never. This is a great opportunity. So yeah, could be.
Yeah, we need more scientific culture also.
Mm, yeah.
It's true. Any of the people that is connected, do you have any questions? Raise your hand some way, switch on your cameras.
Sarah, I have also a curiosity about your patent.
Yes, absolutely.
It was, I mean, I can imagine that all this idea is closely related to your daily life at the lab. So,
Yes, it is.
so how do you differentiate? It was your previous papers and this business. It was very easy to differentiate and to protect only this, and or
That's a very, very good question, and this is it's very difficult. To be honest, I don't think I mastered it. Um, yes, indeed. Sometimes I find myself thinking, well, we did this in the lab. Is it something that can be patented, or should we just go for the publication? And this is the key actually. So what you have to consider when you're thinking that you're going to whether you're going to do a patent or not is if you're bringing novelty. What if your development, your um, product or your science brings novelty to a specific field or for a specific use. And so it has to be very well defined, the novelty and the use of that novelty to be able to do a patent. But of course, I am not to differentiate that. There are some very clear cases. In this case, this was clear because it was a device with specific geometry of microchannels and microfilters that was not that was demonstrating novelty towards a specific use. And so this was clear. But then there are other things like another patent that we have submitted, which is related to the use of nanoparticles to attach to the surface of cells and then pass them through spectrometer and blah, blah, blah. I mean, this is like putting together different things which separate are not novel, but when you put them together, they are novel. And there, it depends on how you sell it, and it also depends on the patent lawyers you have. So when I'm not sure, I speak to our technology transfer office and to our patent lawyers that we have in the institution or the external patent lawyers, and they are usually very much used to identify what can be noble enough to prosecute a patent and not to prosecute a patent. So in doubt, technology transfer office is useful. Yeah.
okay.
Um, I have another question just related to these days, no? Because now it's very difficult, no? With COVID, the hospitals they must work on pandemia and forget about all the other diseases and even cancer, of course. Did you find really much more difficulties to enter your product in the hospitals now, or how it is?
Yes, actually, again, a very good question. Um, what happened is that we last year we got a European project, and we are partners on that European project, and that involves a clinical trial with a collaboration with two hospitals. So we are supposed to receive samples from those hospitals and do the analysis. And then another part of the project is about developing a phone app that is going to read on physical behavior of the patients to identify relapse, combining the objective biomarkers, which is what we provide, the number and phenotypic characterization of CTCs, and the physical changes that a patient might be suffering. So these clinical trials had to be delayed because it was impossible to reach out for the oncologists that were in the project because they are absolutely overwhelmed with all the situation of the pandemic and so on. Also, we had to, after the project was granted, we had to submit approval to the ethics committee of each of the hospitals, and of course, this is delayed because the ethics committee of the hospitals now they have other things to deal with, and they can't do everything at the same time. So the priorities indeed have shifted, and we have had to deal with that. So yes, I mean, it has affected us a bit. Now it seems like it's a bit more into the right path because they know how to deal with things better. We understand this delay, of course, but for other startups, for example, it has opened a window of opportunity because some other startups could pivot what I said in the beginning, their technology to put it at the use of COVID. So uh, we were suggested that to use these for COVID to see if cancer patients were more affected by COVID, for example. But we are a very small startup and we cannot diversify too much. But yes, the pace in the hospitals is completely different. Also, some investors have completely shifted their interests and they want to invest now in viral infections. Mm.
Yeah.
Yes. This
Mm.
which and our opportunities.
Yeah.
Any questions from the audience? Okay. From there, the students, new students. Okay. Have no more questions?
Yes, one.
Yes,
Sergio.
one. Great. Okay, go ahead. Please put the person in front of the blackboard.
Let's go then. You can speak to the computer.
Okay. Hi, I want to know what's the provenience of the investors, if they are public or private?
Yeah. So of our current funding, we have public investment through competitive projects from Portugal, from the European Commission. And then we have two private. One of them is La Caixa Foundation, which is private. We all know La Caixa, right? The bank. They have a foundation that invests in science. We all see the advertisements on the TV. And the other one is Portugal Ventures, which is the largest venture fund in Portugal for science-based technologies. So yes, so it's a combination of public and private funding.
No questions? Billy?
Yes, he at the end, Sergio was thanking you for the lecture.
Oh, thank you very much, Sergio. Thank you all.
Okay. So thank you very much, everybody, for attending. Also, have to thank Marta, which is here with me. Marta. Yeah,
Thank you, Marta.
He's been behind all the time, but I mean, this has been working because she was around and making many trials, going out in of the room of this Zoom.
Yes.
So thank you very much.
Maybe we can have a picture of all the attendees with the camera on and make a picture for the first.
Okay.
It's something that we usually do. Just to
Look at
Yes.
Marta. Marta, congratulations on your PhD. Very nice.
Thank you.
Okay. Yes. Switch on your cameras. Yes.
Okay, and then I am going to. Maybe you come. No, we don't. It's not allowed.
Oh, no. Yes. Okay. So everybody. Other some. Not yet. All the women, huh? You see, all the women switch on the cameras. Huh? By the way, there's two Martas, Sarah. This Marta.
I meant
this
Marta Lafuente.
Marta Lafuente. You know,
Yeah.
you see that.
Yeah. I didn't know her face, but I knew her science. Very good.
That is good. Okay. Hey, Billy, the students can go there to the blackboard. Yes, that's it. That's it. No. No what? Se acaban de ir. Who? Yes,
The security distance. Yeah.
Oh, that's true. Oh. Okay. So let me, because I think the connection is right. Okay. Okay. So say cheese. Cheese with the math and yes. Okay. Yeah.
Nice.
So thank you very much. Muchas gracias,
No,
Sara.
thank you. Gracias a vosotros por invitarme. Ha sido un placer.
Un
Sí.
placer. Y
Nada, cuando
esperemos en
queráis.
la próxima pueda ser en persona.
Sí, eso espero, eso espero. Cualquier pregunta me podéis escribir porque, bueno, eso, sé que hablamos de muchas cosas y que oír así por el ordenador hora y media tampoco es muy guay,
Bueno,
pero
bueno.
es así. Sí.
Ah,
Un placer,
perfecto.
muchas
Un
gracias.
placer. Muchas gracias.
Encantada. Sara, muchas gracias.
Chao, gracias.
Chao.
Have a nice evening. Bye
You too.
bye.
Bye
Bye.
bye.